Hello, this is Rich Whitworth, editor of Biopharm International, and I'm here again today with Anjan Seltz, who's the co-founder and CEO of Phenox Biotech. In the last podcast, we talked about candidate selection, and we also talked about some of their partner decisions and how they've moved forward with their development. Today, we'll be talking a little bit about quality by design approaches that they used and how they've been working with the regulatory authorities. So, Anja, welcome back.
Good morning, Rich. Good to talk to you again.
We can go straight into a little bit of the Phenox approach to quality by design, and perhaps how regulatory expectation matches up with the reality.
Yes, within the regulatory expectations, it's not always that clear what they would expect from you, because these guidelines, they probably give you an overall approach to a certain topic, and then you have to find out, either by experience or by asking someone else with experience, how to do it. And if you then go into discussions with your partners who do these scientific jobs, and you have to either channel their activities, or you have to draw a picture in the end that satisfies the regulatory expectations. So, for example, if you go into developing specifications for your product, the regulatory expectations, as we believe it is within the European Medicines Agency, is that you look at your batches of your product, you look at the reference product, and out of this you derive some specifications for your product. And out of these results, you then justify your specifications, and you probably adjust your specifications through a circle of continuous production of new batches and getting more data on your product. But the development realities are a bit different. The development realities, to say it shortly, someone wants to do a biosimilar, he goes into the published data about the originator product, he takes these specifications, he does a cell cloning, he puts up his cell bank, he puts up his upstream and downstream processing, and then he looks whether it matches or not. So this is the development reality, but actually, if you want to do it in a scientifically sound way in regards to regulatory authorities' expectations, you should do it a little bit different. The result will be the very same, and finally it is just a question of how you organize your project in order to come to the result you would expect and the result the regulatory authority would expect.
Okay, and you mentioned you've been dealing with the EMA, but have you also been working with FDA at all? And if so, do their approaches differ in any way, or are they following a very similar line?
Well, we have started with the EMA, but we have started even earlier than going to the EMA first. We first, when we developed our non-clinical and clinical development plan, we had all set it up. We first went to in kind of a training session we did with the national authority. We said, okay, our team and our biotech partners team, we have not that much experience yet with such activities, so first we go and burn our fingers with the national authority before going to the European authority. And we went into this exercise with the national authority, and it was very helpful. It was helpful for us to understand how such authorities would work like, and also it was very helpful for us because there was the one or the other big hole in our development plan, and they would help us filling it up before we went to the overall European authority. So then we went well prepared to the European authority, and we made a proposal to them in regards to product development, and we agreed on many points, and some points they said, well, you should go a bit in this direction or that direction instead of the direction you had proposed. And we didn't argue much about this because it was all these were all recommendations which were scientifically well-founded, and we could all accept this. But our product shall not only go to market in Europe, it shall also go to market in the USA. So we had the chance that even though the biosimilars guidelines in the USA are not clear yet, in regards to certain biologics, the situation is quite clear, because historically certain biologics have been approved as drugs instead of biologics, such as FSH as well. So we do not fall in this unclear picture of the biosimilars regulation in the USA. So when we addressed the FDA, we first made it clear that we fall in this different category, and not in the biosimilars category, but more on the generic side, even though we are a biologic with our Folia product. When we went to the FDA, let's say before going to the FDA, a lot of people told me, well, the FDA, that's the real regulatory authority, and kind of drew a scary picture. And
right.
When we then actually were in discussions with these people, we found out they are actually the very same as with any other regulatory authority we would have met previously. These people, their first concern is about patient safety out there in the market, and actually they share this concern together with us, with the industry, because only safe products will be a market success. So basically there is nothing to be scared about. It's just about discussing a way to find a good solution to your individual product in order to be approvable and make it attractive in the market. So what we did is the very same as in Europe as well. We drew up the questions we wanted to address, overall the non-clinical and clinical development, but especially also in regards to CMC issues. We addressed these questions to the FDA, always together with a proposal how we would solve the issue. And we went there, and a lot of these issues, they just agreed without any discussion, and certain discussions happened. And what was a really good experience with the FDA was, in our initial discussions, they would kind of overload our development program, such as we would not go into this market anymore. And we signaled them that this was too much. And so by giving us that much to do, they would never achieve their second goal besides patient safety. In regards to biosimilars, they have a second goal, which is bringing affordable medicine to the patient.
Right.
And so we kept discussing with them. We didn't agree. We said, no, we don't agree with the FDA. And they were actually quite, I believe they were quite happy someone would at least tell them, no, we don't agree with you.
Oh, really?
And, yes, we had the impression they don't have much experience of people saying no to them. So anyway, we said no, and we asked them, kept asking them if there were not other ways around. And it was a process during about 1.5 years that we have conducted now together with them, and we kept up discussing with them, and we kept up going back and forth and back and forth. And in the end, now both parties have agreed to a plan that would satisfy all parties' expectations. It would satisfy the FDA's expectation in regards to patient safety and bringing affordable medicines to the patient. And actually, these are the very same goals that we pursue, and we have found agreement. And now we follow it precisely.
Right. And do you think that's something that you've learned for this first candidate that you're describing? Do you think what you've learned during that process would be used for a second candidate, say once you've marketed Afolia and you want to move on to another biosimilar, or is it very much a case by case basis, and that this process of going backwards and forwards is going to happen no matter what, and it's just something that you have to account for?
Well, with all regulatory authorities we have been addressing, we have made this experience that probably in the first discussion they will ask you to do too much in your view. Not in the authorities' view, but in your view, because in the end it still needs to be a reasonable business case you can run. And if they ask you to do too much, probably your business case ain't reasonable anymore. So we have had this going back and forth game practically with all the authorities we have been discussing. And when we signal to the authorities that yes, we are ready to do more than we would have proposed initially, but probably not as much as they would ask us to do, or doing it some other way around than the way they had proposed, they were open for discussions. And I would apply this again to another product development as well. I believe these authorities, they share the industry's interests basically, and so it's nothing to be scared about, and you shall engage in discussions. But one very important thing I believe there is also with authorities, once you have done agreement, you should really stick to it.
Right.
I believe these agreements are like a cooking recipe, and you have to follow it precisely in order to get a good meal on the table, because it will be the very same people who will be measuring yourself again when you go back with your dossier, and they will pull out of the drawer again the agreement you have done a couple of years ago, and they will compare whether you have satisfied all requests in the agreement done previously.
Right, and you mentioned before that you think it's a good idea to choose demanding investigators as well in these regulatory authorities. Is that part of the same process that you can ensure that the recipe, if you will, that you end up with is the right one?
Well, the demanding investigators, the idea about choosing demanding investigators is the credibility topic I would shortly address. Phenox is a no-name company, and as a no-name company, what is very important, be it when serving your first client, which are the regulatory authorities, being it at the time you serve your second clients, which are the prescribing physicians, what you want to transport what you want to bring over in your message is credibility. So if you go the easy way, it will be difficult to keep up with credibility. If you, for example, go to an authority and they say, look, instead of having an 80% statistical power in my clinical trial, which is the common powering of such studies, I will go for a 90% powering, which means more patients, which means more data, which means more credibility. Then these authorities are happy, and I'm sure the prescribing physician will be happy as well to, when he has the choice of using a proven product, a product he has used for 10 years, 20 years, I don't know, or he has to switch for another product, there need to be strong reasons he would switch to another product. And credibility is one central issue. So demanding investigators is just one part of building up credibility for your product.
I see. And perhaps we can talk a little bit about one big question in biosimilars is how similar is similar, something I've heard in a number of different presentations. So maybe you can tell us a little bit about that and your process and how far you went in your sort of comparability exercises and so on.
How similar is similar? I must admit, I don't know, because we will only know once we have submitted our data to the authorities whether they accept the similarity approach. But basically, it's very difficult again if you look at the guidelines, at the European guidelines which describe the similarity requests for such products. They are kind of generally held. It's not stated what kind of test you should do or not. But what we found out in discussions, be it with peers in the industry, be it with the authorities, we found out that definitely just running your release analytics, for example, with your product and the reference product definitely ain't enough. You will definitely have to do it on drug substance level, you have to do it on drug product level, and then the set of methods needs to do a full protein characterization. And the full protein characterization, that's the big question. What means full protein characterization? And our choice was to first do the easy ones, all the methods that everybody would do. And when we had all this done, let's say in parallel that we were doing this, we were investigating with people who might know about this, who probably have already brought some biosimilars through to approval. We were talking with such experts, and they were giving us more hints. And in the end, we ended up with a battery of test methods of more than 20 tests that would, we believe, fully characterize the protein. It goes up to the tertiary structure, glycosylation patterns, whatever you may have. Scientists know better than I do, but it's very difficult to find out, and there definitely is a case-by-case situation for the one or the other product. Comparability exercises is definitely key to approval.
Yeah, so you think, I guess you do, in that case, you do as much as is possible, essentially. I know you said in your presentation you need to go one step further than what perhaps the regulators expect, and certainly, I guess, with a smaller biologic like FSH, as you mentioned, I guess you can really run the whole gambit of tests, whereas a monoclonal antibody is obviously a lot larger and a lot more complex, and I would imagine it was a lot more difficult to do. So, yeah, I guess the idea of going one step further is key there.
Going one step further is, again, this topic of building up credibility. And in all aspects of product development, we would agree with the authorities on doing something, and then we would try to do this something, but a step further than only this something, just in order to show the authorities, okay, look, we want to go to market, we will bring enough reasons for you to give us an approval. And the very same philosophy we apply to the comparability exercise as well.
Okay, well, thank you very much for your insight on the regulatory side, and hopefully we'll get another chance to talk to you in the near future about perhaps some of the differentiators you mentioned for your compound going forward. So thank you very much for your time, and we'll speak to you again soon. Thank you.
Thank you.